Thank you. So I'm a final year PhD student here in Oxford in the groups of Michael Booth and Thomas Mill. And this is not your standard nuclear targeting moieties, has been largely explored in the ASO field, slightly different, so bear with me. Nucleic acid therapeutics, they come in very many flavors. There are drugs on the market. They've got very powerful applications in biomedical technologies of all sorts. And antisense oligonucleotides win the race with the largest number of approved drugs on the market, with some targeting sort of a variety of different indications, a lot in sort of the CNS space, with drugs approved last year. And this is eplontersen, which has the same sort of GalNAc moiety that's featured in siRNA. And people have gone over the mechanism of action, so I'm not going to spend too much time on it. But broadly speaking, the two main classes of antisense oligonucleotides, their mechanisms of action can be roughly broken up into either splice modulation, which is where the backbone chemistry is so exotic it can no longer be recognized by the enzyme. So it essentially just sits on your target pre-mRNA and affects sort of the transcription or the splicing outcomes, essentially, to allow for sort of functional incorporation there. Or you have the more sort of traditional enzymatic recruitment, if you will, where your DNA ASO binds to your target RNA. That DNA RNA duplex is then recognized by the enzyme RNase H, and you see actual mRNA knockdown. So like others in the area, we had a very sort of simple question. It's quite well documented that the main mechanisms of actions occur predominantly within the nucleus, in that splicing machinery was within the nucleus, and RNase H has the highest effective local concentrations within the nucleus. We wondered whether we could use a small molecule importer, which has, a lot of different advantages over sort of cell-penetrating peptides or NLS motifs, if you will, to achieve the same outcome, to improve sort of the localization of ASOs within the nucleus. And we were inspired by this work from Stuart Schreiber's lab at the Broad, which designs this bifunctional, essentially, which comprises of a favorite molecule, JQ1, that binds bed bromodomain protein. So essentially, it's a very promiscuous bind in that it hits a lot of different bed bromodomain proteins. And they have shown that they can use this mechanism that bed bromodomain proteins have, where they sort of shuttle between the nucleus and the cytoplasm to carry out sort of different functions, to bring GFP just as a test system, but they'd show with loads of other different molecules from like this more sort of diffuse profile in the cytoplasm to a more concentrated profile within the nucleus. So they can get sort of import of this cytosolic protein into the nucleus. And so we decided to put JQ1 on an ASO and see if a similar mechanism could be expanded to ASO nuclear localization. And this is just click chemistry. We love click chemistry, beautiful chemistry, very great yields, super pure conjugates, great. And then we took it to a similar model that Debashish also showed you work in, which is this sort of defective luciferase gene model in HeLa cells, where in the absence of this splice switching oligonucleotide, you get, defective luciferase. You don't get any sort of luminescence. But when you treat it with your oligonucleotide, this is the sort of baseline luminescence profile that you see. And with the addition of that one JQ1 moiety, we could already see a twofold increase in activity at all the different sort of concentrations that we tested. And this gave us confidence that this one simple single covalent modification could improve the splice switching activity of oligonucleotides by over 2 times. We're like, okay, this is a dummy system, dummy reporter gene. We decided to go after a more interesting target, which is essentially oblamersen. This is another just chemistry. So same chemistry, same workflow, expandable to different backbones. We made a different conjugate, now targeting BCL2. So oblamersen, or G3139, was an ASO chemosensitizer that failed in phase 3 clinical trials. So it made it almost through to the end. And the reasons that were cited was sort of poor uptake or poor cellular compartmentalization, essentially. And we thought, okay, can we make this drug better? And indeed, that is the case. So through that one simple JQ1 modification, we see an increase in, mRNA knockdown with RT-qPCR, which we also track with the Western blot. visible at the protein level. And again, we see a concentration-dependent increase at both the RNA and the protein level. So we're able to improve the activity of this one almost therapeutic through this one very simple covalent modification by over 2 times. We then decided to ask, what is the mechanism of action through which this conjugate is functioning? For this, we did a very simple blockade assay, and the principle here is if this JQ1 ASO conjugate binds to the same sort of molecular protein targets as the small molecule, in the presence of excess amounts of the small molecule, it would be outcompeted for activity. So that's the baseline activity that we see in the absence of, no small molecule. And this is the baseline activity of the ASO in the presence of excess amounts of just a small molecule. And as expected, we don't, want to see any sort of differences here. But when you treat it, when you take the JQ1 ASO conjugate, already you can see a sort of rescue in terms of the ability of the ASO to knock down, and that's completely lost at higher concentrations of excess JQ1. So through this very sort of simple assay, we could show that in the presence of excess amounts of small molecule JQ1, the JQ1 ASO conjugate could no longer sort of co-opt into the same mechanisms and carry out its function. So that gave us confidence that indeed the ASO JQ1 conjugate is able to bind to the same sort of molecular targets as the small molecule alone. We then wanted to see, okay, are we actually seeing nuclear accumulation? I'm not trained in microscopy. I'm doing these experiments now. But we thought to do a very sort of simple fractionation assay. So for that, we designed a different conjugate where we put a dye on. And the first thing to check is obviously whether the addition of the Cy3 moiety has any effect on the knockdown that we're seeing. And indeed, that's not the case. We still see significant increase in knockdown with the JQ1 and the Cy3. And then I go on to do a very sort of simple fractionation experiment where you first collect the cytoplasm, probe for GAPDH, which is cytoplasmic protein. Then you pick the nuclear fraction, probe for, menin, which is a very beautiful transcription factor. Probe for that. We see clear sort of partitioning. And then we look just through a plate reader at the Cy3 fluorescent signal to show in terms of where the ASO is partitioning baseline with time. So by 24 hours at that high concentration of 0.5 micromolar, you're seeing a fair amount of signal in the nucleus. But when you add the JQ1 moiety, you can see an almost doubling of the amount of ASO within the nucleus and almost a 50% partitioning at 0.5 micromolar at the 24-hour time point. And so this presented a more sort of quantitative link in our eyes that the improvement in activity that we were seeing was because of an enhancement in nuclear localization. And so I hope with that work, I've been able to show you that something that's been debated within the field for a really long time as to whether an increase in nuclear localization actually does have impacts on activity and outcome. And we think this is a very simple, elegant, easy way to show that that is indeed the case. It's a simple one-step chemical modification. I've done it with multiple different targets. I've gone after MALAT1. I've done the work in HEX, in HeLa, in A549s, in AML cell lines. It's super robust, loads of different targets, loads of different backbone chemistries, which is exciting to see. We go after the 2 different main mechanisms of action as well, which is splice switching and RNase H-mediated knockdown. We can make almost drugs better, which is always a win, right? Take candidates that almost made it, cross the finish line. And I think my PhD, we've been working in this area really to start thinking about small molecules like JQ1, for example, as just effectors of the intracellular fate of nucleic acids or biologics of different kinds. And I won't talk about it today, but other work in my PhD that I've done is about showing that you can actually achieve knockdown of your target mRNA in the lysosome or in the cytoplasm instead, again, through a small molecule ASO conjugate. And I've also designed oligoproteins, so oligonucleotides linking proteins together and showing that you can actually use very simple principles in DNA nanotechnology to turn oligoproteins off instead of, the more traditional protein activation prodrug strategies, essentially. And obviously, no, all science takes a village, and I'd like to, thank my supervisors and my funding agencies and all of you for your attention. Thank you.